Hello all, this is Priyadharshini, Assistant Professor, Department of Biotechnology, JJ College of Arts and Science, Pudukottai. Today we are going to discuss about the sequence analysis, which is a topic from bioinformatics. And these are all the content today we are going to see superficially: introduction, DNA and protein sequencing, how to assemble the sequence and how to align the sequence, and multiple sequence alignment, demonstration of protein sequencing and its applications, and last we are going to see the references where we have taken all these things. Sequence analysis. And sequence analysis is the process of subjecting a DNA, RNA, or a protein sequences to a wide range of analytical methods. We are doing the sequence analysis to understand the features, function, structure of the particular sequences. And it can be done by using sequence assembly analysis and by searching the databases. First of all, let's see what is DNA sequencing. DNA sequencing is nothing but we are going to identify the order of nucleotides, adenine, guanine, cytosine, thymine in the strand of the DNA. Likewise, in case of protein sequencing, we are just analyze the amino acids, number of the presence of amino acids in the sequences. Next one is sequence assembly. In case of sequence assembly, we have to go for some steps. First, we have to cut the DNA into small pieces, and then we will get some individual small fragments. Each individual fragments has to be sequenced. At last, we have to reconstitute into the original data DNA by merging the informations on various fragments. Let's see sequence alignment. In the sequence alignment, uh, usually it is of two types. One is pairwise alignment, another one is multiple sequence alignment. Pairwise alignment means if you are comparing the two sequences, means it's pairwise alignment. Multiple sequence alignment means if you are comparing more than two sequences, that comes under multiple sequence alignment. Why we have to do the sequence alignment means to find out the similarities between the sequences, whether we are finding out the similarities between the known with the unknown, or we can compare five known sequences together. And this is the format of multiple sequence alignment. I have said you that multiple sequence alignment is referred to the process or the result of sequence alignment of three or more biological sequences, generally protein, DNA, or RNA. Here we can use cholesterol W, cholesterol X, BLAST, FASTA, and FIRE2. Any of the tool we can use for doing multiple sequence alignment. And here we are going to see about the demonstration of the protein sequencing. Usually by the means of a chemical degradation method, we will be going for advanced degradation method. Here we have given the name of the protein, that is human prion protein. uh, Human prion protein in NCBI, that is the NCBI that acts as a interface between the user and the data banks, and we can retrieve the human prion protein from the data bank, and we can save it into the computer. And then second step is we have to open the blast page and paste the sequences which we have retrieved from the data bank and make it run. It will give you some of the 10 sequences which is similar to our sequences. Our sequences means the sequence which we have given as a input is our sequences. So the 10 sequence that may be similar to our sequence. How the 10 sequence that is coming out from the blast is, we have to look for the presence of the conserved domain, and then after retrieving the 10 sequences from the data bank, we have to construct the phylogenetic tree. By means of phylip software or cluster W, we can construct the phylogenetic tree, and then we have to predict the secondary structure of the human prion protein. And this is the website for NCBI, that is National Center for Biotechnological Information. Here see, they have selected the protein, and next to that we have to write the protein name. We can get the data, and this is the blast page. Here we have to paste that sequence, and we have to go for the option blast, and this is the cluster omega software for the multiple sequence alignment. And the Pyri 2 is a software which we can use for the prediction of the secondary structure, and also to view the three-dimensional view of the human prion protein. You can predict the secondary structure of the protein by using the software SOPMA. For the tertiary structure, we can use this Pyri software to determine the three-dimensional view of the particular proteins. And here they have listed out the some of the applications of this sequence analysis. The comparison of sequences in order to find out the similarity, often to infer if they are related. Homologous means the sequence with they are related. And then identification of the intrinsic, that is inner feature of this sequence, such as active sites, post-translation modification sites, gene structures, distribution of introns, exons, etc. And for sequence differences and variations in case of mutations and SNPs for the genetic marker, and revealing the evolutionary relationship between them, and to identify the molecular structure of the sequence, and to for the genetic disease also. This what is the applications, and these are all the references for the particular presentation. Thank you.